In this video, we'll cover everything within the infection and response topic, from how pathogens cause disease to how the body defends itself, and how medicines and vaccines are developed to protect us. Today, we're learning about various disease causing organisms and their distinctive characteristics, how these organisms make us ill through different mechanisms, the various transmission methods that allow infections to pass from one host to another, and finally, how these infections affect both the plant and animal kingdoms. Disease causing microorganisms, known as pathogens, invade the body and trigger illness. These microscopic invaders come in four main categories: bacteria, viruses, protists, and fungi. Each operates differently when causing sickness. Bacteria are microscopic single celled organisms. Despite their tiny dimensions, they multiply quickly once inside the human body. What makes bacterial infections problematic is that many bacteria release harmful chemicals as they grow. These substances interfere with normal cell function and harm tissues, resulting in the symptoms we experience during infection. Viruses are considerably smaller than bacteria, approximately 1/100th of a bacterial cell's size. Unlike bacteria, viruses lack cellular structure consisting merely of genetic material encased in protein. Unable to multiply independently, viruses hijack the host cell's internal mechanisms. They force our cells to create viral components before assembling numerous new virus particles. Eventually, the infected cell typically ruptures, releasing newly formed viruses that infect neighboring cells. This destruction explains many symptoms associated with viral infections. Moving to protists, these are more complex, single celled organisms containing a nucleus. Most disease causing protists function as parasites residing on or within hosts while causing cellular disruption. Many protist infections reach humans through carrier organisms called vectors. These carriers transport the pathogen without becoming ill themselves. Our final pathogen category, fungi, ranges from single celled organisms to complex structures formed of thread like filaments called hyphae. These filaments can extend into human skin or plant surfaces, establishing infection. Many fungi distribute through reproductive particles called spores, enabling the infection to spread between hosts. Regarding transmission, pathogens spread through three primary routes. The first involves water, where organisms contaminate drinking sources or recreational waters. For instance, certain bacterial infections occur when people consume water containing waste from infected individuals. Airborne transmission forms our second route. Some pathogens travel through air currents, particularly in moisture droplets released during respiratory actions like coughing. Respiratory infections commonly spread this way. Our third transmission method involves direct contact. Numerous pathogens transfer through touching contaminated objects or infected individuals. Many skin infections propagate through shared surfaces in communal areas. It's worth noting that pathogens don't exclusively target humans. Plants and animals also suffer from various infections. These can significantly impact growth, reproduction, and survival across all living kingdoms. Today, we're learning about three viral diseases: measles, HIV, and tobacco mosaic virus, then looking at two bacterial infections, salmonella and gonorrhea, studying a fungal disease called rose black spot, and finally investigating malaria, a disease caused by a protist. Let's start with viral diseases. Measles is a viral infection that causes fever and a distinctive red skin rash. It spreads through droplets in the air when an infected person sneezes or coughs. These tiny moisture droplets can be inhaled by others nearby. People with measles typically develop a high temperature and a red rash that starts on the face and spreads across the body. Measles can be serious or even fatal if complications develop. It can lead to pneumonia, which is inflammation of the lungs, or encephalitis, which is inflammation of the brain. Because of these serious risks, most children receive vaccination against measles when they're young, which has helped reduce cases worldwide. The second viral disease we'll examine is HIV, which stands for Human Immunodeficiency Virus. HIV spreads through sexual contact or by exchanging bodily fluids, such as blood. This often happens when people share needles during drug use. When first infected with HIV, a person may experience flu like symptoms for a short period. They might then have no symptoms at all for several years. During this time, however, the virus continues to attack the immune system. It specifically targets immune cells, gradually weakening the body's natural defenses. Without treatment with antiretroviral drugs, the immune system becomes severely damaged, leading to AIDS, which stands for Acquired Immune Deficiency Syndrome. At this stage, the body cannot fight off other infections or cancers effectively. People with AIDS often develop infections that wouldn't affect someone with a healthy immune system. Viruses don't just affect humans, they can infect plants too. Tobacco mosaic virus, which is also known as TMV, affects many plant species, including tomatoes. It causes a distinctive mottled pattern on the leaves where some areas appear discolored. This pattern gives the virus its name. The discoloration means the plant can't photosynthesize properly, which affects growth. Infected plants often produce fewer fruits and may be stunted. TMV spreads when plants come into contact with each other or through gardening tools. Unlike animal viruses, it doesn't need vectors like insects to spread. Now let's move to bacterial diseases. Salmonella is a type of bacteria that causes food poisoning. People infected with salmonella experience fever, stomach cramps, vomiting, and diarrhea. These symptoms usually appear within a day or two of infection. These symptoms occur because the bacteria produce toxins that irritate the digestive system. You can get salmonella by eating food contaminated with the bacteria. This includes undercooked meat, eggs from infected birds, or food that's been cross contaminated during preparation. In the UK, most poultry are vaccinated against salmonella to help control its spread. This public health measure has reduced human infections significantly. The second bacterial disease we'll look at is gonorrhea, a sexually transmitted infection. Unlike salmonella, gonorrhea spreads through sexual contact, especially unprotected sex. A person with gonorrhea will often feel pain when they urinate. Another common symptom is a thick, yellow, or green discharge from the affected areas. Gonorrhea used to be easily treated with penicillin. However, many strains of the bacteria have become resistant to this antibiotic. This antibiotic resistance is a growing concern in treating bacterial infections. It happens when bacteria evolve in response to the drugs used against them. To prevent gonorrhea spreading, people can use barrier methods of contraception like condoms, and those infected can be treated with appropriate antibiotics. Now let's take a look at fungal diseases. Rose black spot is a common fungal disease affecting rose plants. This fungus causes purple or black spots to develop on the leaves of rose plants. The spots have irregular edges and can grow quite large. As the disease progresses, affected leaves often turn yellow and fall off prematurely. This early leaf loss weakens the plant. When the plant loses its leaves, it can't photosynthesize as effectively. This means less energy for growth and flowering. The fungus spreads through the environment by water or wind. Wet conditions particularly favor its development. Gardeners can treat rose black spot with fungicides and by removing affected leaves. Good air circulation around plants also helps prevent infection. Finally, let's examine malaria, a disease caused by a protist parasite. The malaria protist has a life cycle that involves mosquitoes acting as vectors. Female mosquitoes spread the disease when they bite humans. When an infected mosquito bites a person, it injects the parasite into the bloodstream. The parasite then travels to the liver and begins multiplying. Malaria typically causes recurring episodes of fever, along with chills, sweating, and headaches. These fever cycles correspond to stages in the parasite's life cycle. These episodes can be very severe and, in some cases, fatal. Malaria is particularly dangerous for young children and pregnant women. The spread of malaria can be controlled by preventing mosquitoes from breeding. This involves eliminating standing water where mosquitoes lay their eggs. Using mosquito nets while sleeping also helps avoid being bitten, as does using insect repellent. In areas where malaria is common, people may take anti-malarial drugs to prevent infection. These medications help stop the parasite from developing in the body. Understanding these disease examples helps us appreciate how different pathogens affect living organisms in various ways. It also highlights the importance of prevention. Today, we're learning about the body's non-specific defense mechanisms, the barriers that stop pathogens entering in the first place, then examining what happens when pathogens breach these barriers and enter the body, and finally exploring how our immune system specifically targets and destroys different types of pathogens. Our body has evolved multiple layers of protection against disease. The first line of defense involves preventing pathogens from entering our bodies at all. The skin forms an effective physical barrier against pathogens. It provides a tough, waterproof covering over most of our body. When intact, it's very difficult for microorganisms to penetrate this barrier. Our skin doesn't just provide a physical barrier, it also produces antimicrobial substances that kill many pathogens on contact. This chemical defense adds another layer of protection. Inside our nose, we have tiny hair like structures and mucus that trap particles from the air we breathe. This prevents many potential pathogens from going deeper into our respiratory system. Similarly, the trachea and bronchi, the airways leading to our lungs, are lined with cilia, microscopic hair like projections. These tiny structures move in a coordinated way to waft mucus upwards to the back of the throat, where it can be swallowed. This mechanism is sometimes called the mucus escalator because it continuously moves trapped particles away from our lungs towards our digestive system. Speaking of our digestive system, the stomach produces hydrochloric acid, which creates a very low pH environment. This acidic condition kills many pathogens that might be present in our food or drink. But what happens if, despite all these defenses, pathogens still manage to enter our body? This is where our immune system comes into play. If pathogens breach our external defenses, our body activates internal defense mechanisms. White blood cells are the most important part of this immune response. White blood cells circulate in our bloodstream and can move into tissues throughout the body. They constantly patrol for foreign invaders, and when they detect pathogens, they respond in several ways. One method white blood cells use is called phagocytosis. In this process, certain white blood cells surround and engulf foreign organisms, then digest them using enzymes. This is like the white blood cells eating the pathogens. Another response involves the production of antibodies. When white blood cells encounter a foreign substance called an antigen, they produce specific proteins called antibodies. These antibodies lock onto the specific antigens on the pathogen's surface. This tagging helps other white blood cells identify and destroy the invader. It also prevents the pathogen from attaching to our body cells. If a person is infected with the same pathogen again, their white blood cells will rapidly produce the same antibodies. This quick response often prevents the disease from developing. We say the person has become immune to that specific pathogen. The third way white blood cells defend against pathogens is by producing antitoxins. These substances neutralize the toxins released by certain bacteria, preventing them from causing harm to our cells. Our defense systems work together to protect us from the countless pathogens we encounter daily. Without these mechanisms, even minor infections could become life threatening. Let's begin by going over exactly what we'll cover in this video. First, we'll examine how vaccination works to protect individuals from disease. Next, we'll look at how vaccinating a large portion of the population helps reduce the spread of pathogens. Finally, we'll discuss some of the benefits and limitations of vaccination programs. When you're infected with a pathogen, your immune system takes time to recognize the threat and mount a defense. During this learning period, you can become quite ill as the pathogen multiplies in your body. Vaccination works by preparing your immune system in advance without you having to suffer the actual disease. Vaccines typically contain dead or inactive forms of pathogens. These cannot cause the disease but still carry antigens, the molecular structures that trigger an immune response. When these weakened pathogens are introduced into your body, usually by injection, your white blood cells recognize them as foreign. This stimulates your immune system to produce antibodies that specifically target those antigens. For example, the MMR vaccine contains weak versions of the viruses which cause measles, mumps, and rubella. Each component triggers the production of specific antibodies against that virus. The clever part happens if you later encounter the real, active pathogen. Your immune system remembers the antigens from the vaccination and can rapidly produce the right antibodies to destroy the pathogen before it causes illness. This rapid response is what makes vaccination so effective. Your body doesn't need to go through the learning phase again. It's already prepared for that specific threat. Vaccination doesn't just protect individuals, it also helps protect entire communities through a concept called herd immunity or population immunity. When a large proportion of a population is vaccinated, pathogens cannot spread easily because they encounter fewer susceptible individuals. This creates a form of indirect protection for those who aren't vaccinated. This is particularly important for protecting vulnerable groups who cannot be vaccinated, such as very young babies, people with certain medical conditions, or those with severely weakened immune systems. Vaccines have had remarkable success in controlling once common diseases. In the UK, diseases like polio, measles, whooping cough, rubella, mumps, and tetanus have become rare thanks to widespread vaccination programs. Some diseases have been impacted even more dramatically. Smallpox, once a devastating global disease, has been completely eradicated worldwide through vaccination. Polio infections have fallen by 99% globally. Large outbreaks or epidemics can be prevented when enough people in a community are vaccinated. This breaks the chain of infection, as the pathogen cannot easily find new hosts to infect. However, if vaccination rates drop below certain thresholds, diseases can quickly resurge. When unvaccinated people are clustered together, pathogens can spread rapidly through these groups. It's important to note that while vaccines are extremely effective, they're not perfect. Some vaccines don't provide complete immunity for everyone who receives them. The protection may vary based on factors like age and overall health. Very rarely, people may experience side effects from vaccines. Most of these are minor and temporary, like soreness at the injection site or a mild fever. Serious reactions are extremely uncommon. In summary, vaccination works by safely exposing our immune system to pathogen antigens, allowing it to develop a rapid response if it encounters the real pathogen later. This not only protects individuals, but also reduces the spread of disease through populations. By understanding how vaccination works, we can appreciate why high vaccination rates are crucial for maintaining public health and preventing the resurgence of dangerous diseases. In this video, we'll cover three key topics: how painkillers manage symptoms without curing disease, how antibiotics fight bacterial infections, and why using these medications correctly is so important. When we're ill, medications can help in different ways. Some drugs treat the symptoms of illness, while others actually tackle the underlying cause. Painkillers, such as aspirin, are drugs that relieve pain, which is certainly helpful when you're feeling unwell. However, it's important to understand that painkillers don't actually tackle the cause of the disease or kill pathogens. Similarly, many over the counter medicines for colds and flu work by reducing symptoms like congestion, coughing, or fever. These cold remedies make you feel better, but don't actually fight the virus causing your cold. Antibiotics, on the other hand, work in a completely different way. Drugs like penicillin actively fight against bacterial infections by either killing bacteria or preventing them from reproducing. This is a crucial distinction. Antibiotics don't just make you feel better, they actually help eliminate the cause of the disease when that cause is bacterial. Different antibiotics are effective against different types of bacteria. This is why it's important to be treated with the right antibiotic for your specific infection. A key limitation of antibiotics is that they only work against bacteria. They have no effect on viruses that cause diseases like colds or flu. This is because viruses have a completely different structure and life cycle compared to bacteria. Viruses reproduce inside our body cells, which makes it very difficult to develop drugs that can destroy the virus without also harming our own cells. This is why many viral infections are treated by managing symptoms while our immune system fights the virus. The discovery and development of antibiotics has been one of the greatest medical advances of the 20th century. Before antibiotics, even minor bacterial infections could be life threatening. Since their introduction, antibiotics have greatly reduced the number of deaths from bacterial diseases. Infections that were once commonly fatal can now be effectively treated with a course of antibiotics. To summarize, painkillers and other symptom relieving medications help us feel better by treating the symptoms of disease. Let's begin by going over exactly what we'll cover in this video. First, we'll examine the historical origins of medicines and how traditional remedies influenced modern pharmaceutical development. Next, we'll investigate initial laboratory evaluation methods for potential new treatments. Then we'll look at how promising compounds are assessed in controlled human studies. Finally, we'll consider the importance of scientific verification before medications receive approval. Throughout history, the natural world has provided many therapeutic substances. Plant species synthesize various chemical compounds as defense mechanisms against insects and microorganisms. Remarkably, some of these natural substances can effectively treat human ailments. For centuries, people observed which plants seemed to offer medicinal benefits. These observations formed the foundation of traditional healing practices worldwide, many of which eventually led to contemporary pharmaceutical products. Consider aspirin, widely used for pain relief and fever reduction. This common medication originated from compounds identified in willow bark. Long before modern pharmaceutical science, people recognized willow's pain relieving properties. Similarly, the cardiac medication digitalis was developed from compounds present in the foxglove plant, which had historically been used for heart ailments. Microorganisms have also yielded important medicinal compounds. A noteworthy example comes from Alexander Fleming's laboratory work. While examining bacterial cultures, Fleming observed a curious phenomenon. A mold contamination had created a bacteria free zone in one of his culture dishes. This mold, identified as penicillium, was producing a substance that eliminated the surrounding bacteria. Fleming had discovered penicillin, inaugurating the antibiotic era in medicine. In contemporary times, pharmaceutical manufacturing occurs on an industrial scale. Although many medications still derive from natural substances, chemists frequently modify these compounds to enhance effectiveness and minimize unwanted effects. Before any medication becomes available to patients, it undergoes extensive evaluation through multiple testing phases. This rigorous assessment process ensures both safety and efficacy. Laboratory evaluation represents the first phase of this process. During this stage, researchers examine how compounds interact with human cells and tissues in controlled laboratory conditions. These experiments provide preliminary insights into potential therapeutic mechanisms. However, laboratory studies using isolated cells cannot fully predict how substances will behave in complete biological systems. For example, assessing cardiovascular medications requires evaluation in organisms with functional circulatory systems, not just isolated heart cells. Following successful laboratory studies, researchers conduct animal studies as part of preclinical evaluation. These investigations serve multiple purposes. They assess therapeutic effectiveness, identify potential toxicity issues, and help determine appropriate dosage ranges. UK legislation mandates testing proposed medications on two different mammalian species before human trials begin. This requirement generates significant debate. Some advocate for animal testing as a necessary safety measure, while others question its ethics and seek alternative methods. Compounds that successfully complete preclinical evaluation advance to human clinical trials, which proceed through several distinct phases. Initially, researchers administer experimental medications to healthy individuals. These early trials focus primarily on safety assessment rather than therapeutic benefit. Researchers begin with minimal concentrations of the compound, gradually increasing amounts if participants tolerate the substance well. When safety data from healthy individuals appears favorable, researchers then evaluate the medication in patients with the targeted condition. These studies determine optimal therapeutic concentrations, amounts that maximize beneficial effects while minimizing adverse reactions. To accurately assess effectiveness, researchers randomly assign participants to different treatment groups. Some receive the experimental medication, while others receive a placebo, an inactive substance appearing identical to the medication under investigation. This methodology allows researchers to distinguish actual medication effects from perceived improvements that occur simply because someone believes they're receiving treatment, the placebo effect. Many clinical studies employ blinding techniques to minimize bias. In blind studies, participants remain unaware whether they've received the actual medication or placebo. Double blind studies extend this principle further. Neither participants nor the immediate research team knows who received which substance until the study concludes. This approach prevents unconscious biases from influencing how researchers evaluate participants or how participants report their experiences. Before publication, research findings undergo peer evaluation. During this process, independent scientists scrutinize the methodology and conclusions to verify scientific validity and procedural integrity. This critical review mechanism helps prevent unsubstantiated claims about new medications and ensures that only thoroughly validated information reaches medical practitioners and patients. Developing new medications involves extensive resources, often requiring many years of research and significant financial investment. However, this comprehensive approach helps ensure that medications reaching patients offer genuine therapeutic benefits with acceptable safety profiles. Let's begin by going over exactly what we'll cover in this video. First, we'll explain what monoclonal antibodies are and how they differ from natural antibodies. Next, we'll examine the specific process scientists use to produce these antibodies. Finally, we'll explore their various applications in diagnosis, research, and treatment. Let's start by understanding what monoclonal antibodies actually are. Antibodies are normally produced by our immune system's B lymphocytes, a type of white blood cell. These Y-shaped proteins bind to specific foreign substances, helping our body identify and neutralize threats. When our immune system responds naturally, many different B lymphocytes produce slightly different antibodies. In contrast, monoclonal antibodies are identical copies that all target exactly the same binding site on one specific antigen. This remarkable specificity makes monoclonal antibodies incredibly useful tools. Because they only bind to one particular target, they can be used to identify or attack very specific substances or cells. Now, let's look at how scientists produce these specialized antibodies. The process begins by stimulating mouse lymphocytes to make a particular antibody. This is done by injecting a mouse with the specific antigen the scientists want to target. However, lymphocytes don't survive long in laboratory conditions. This presented a significant challenge when scientists first attempted to produce antibodies artificially. The breakthrough came when researchers discovered they could fuse these antibody producing lymphocytes with tumor cells to create what's called a hybridoma cell. This hybrid cell inherits two crucial characteristics: the ability to produce a specific antibody from the lymphocyte and the ability to multiply rapidly and indefinitely from the tumor cell. After creating these hybridoma cells, scientists isolate individual cells and allow them to multiply, creating clones. Each clone produces identical antibodies, all targeting the same antigen. The resulting population of identical cells all produce the same antibody. Scientists can collect these antibodies and purify them for various applications. Let's now explore some of the important uses of monoclonal antibodies. One common diagnostic application is pregnancy tests. These tests contain monoclonal antibodies that specifically bind to a hormone called HCG, which is only present in the urine of pregnant women. When a woman uses a pregnancy test, her urine moves along the test strip. If she's pregnant, the HCG in her urine binds to the antibodies, creating a visible line or symbol on the test. In medical laboratories, monoclonal antibodies are used to measure levels of hormones and other chemicals in blood samples. They can also detect specific pathogens, making them valuable tools for disease diagnosis. Research scientists use monoclonal antibodies to locate specific molecules within cells or tissues. By attaching a fluorescent dye to the antibodies, researchers can see exactly where their molecules of interest are located in a sample when viewed under a microscope. Perhaps one of the most significant applications is in treating disease, particularly cancer. Cancer cells often have specific markers on their surface that aren't found on normal cells. Monoclonal antibodies can be designed to recognize these unique markers. These therapeutic antibodies can work in several ways. They might be bound to a radioactive substance, allowing targeted delivery of radiation to cancer cells. Alternatively, they could carry a toxic drug that kills the cancer cells once the antibody attaches to them. Some monoclonal antibodies are linked to chemicals that stop cells from growing and dividing. In each case, the antibody delivers its payload specifically to cancer cells, minimizing damage to healthy tissue. This targeted approach differs significantly from traditional cancer treatments like standard chemotherapy and radiotherapy, which affect many body cells in addition to the cancer cells. Monoclonal antibodies precisely target specific cells, potentially reducing side effects. Despite their usefulness, monoclonal antibodies do have limitations. They can cause side effects that weren't initially expected. When first developed, scientists thought that because they target specific cells so precisely, they would have very few side effects. Additionally, the development of new monoclonal antibody treatments is costly and time consuming. This means they aren't as widely used in treatment as scientists originally hoped they would be. Understanding monoclonal antibodies helps us appreciate how scientific innovations can harness natural biological processes to create powerful tools for medicine and research. These laboratory created molecules continue to play an increasingly important role in diagnosing and treating a range of conditions. In our next episode, we'll explore how plants respond to pathogens and the various defense mechanisms they've evolved to protect themselves from. Let's start by outlining the main points we'll go over. First, we'll look at the common signs that indicate a plant might be diseased. Next, we'll examine the various types of pathogens that can infect plants. Then we'll explore the different methods used to identify specific plant diseases. Finally, we'll investigate the fascinating physical, chemical, and mechanical defense mechanisms plants use to resist infection. Plants, like all living organisms, are susceptible to diseases. Various pathogens can infect them, including viruses, bacteria, and fungi. We've already learned about tobacco mosaic virus and rose black spot in previous episodes. Plants can also be damaged by insects such as aphids, which feed on plant tissues and cause significant harm. When a plant is infected with a disease, it typically shows visible symptoms. Being able to recognize these signs is the first step in diagnosing plant problems. One common indicator of plant disease is stunted growth. Plants may be smaller than expected or grow more slowly than healthy specimens. This happens when pathogens interfere with the plant's ability to produce energy or transport nutrients. Spots on the leaves are another telltale sign of many plant diseases. These spots may vary in color, size, and pattern depending on the specific pathogen. For example, fungal infections often cause distinctive dark spots on leaves. Some plant diseases cause patches of decay or rot on leaves, stems, or fruits. These areas may appear soft, discolored, or water soaked. Plants suffering from certain diseases may develop malformed stems or leaves that appear twisted, curled, or otherwise abnormal compared to healthy plants. Discoloration is another important sign. Leaves may turn yellow, brown, or other unusual colors when the plant is infected. This happens when diseases affect the plant's ability to produce chlorophyll, or when pathogens damage plant tissues. If you're examining plants for potential insect infestations, careful observation often reveals the pests themselves on the plant. Looking at the undersides of leaves or at stem joints can help spot these unwanted visitors. Once you've detected signs of disease in a plant, the next step is identifying the specific problem. There are several methods for this. One approach is consulting gardening manuals or reliable gardening websites. These resources often include photographs and descriptions of common plant diseases, which can help match observed symptoms to specific pathogens. For more definitive identification, infected plant material can be taken to a laboratory where scientists have specialized equipment and expertise. They can examine samples under microscopes and perform tests to identify the pathogen. Modern testing kits are also available that use monoclonal antibodies to identify specific plant pathogens. These work similarly to the testing kits we discussed in our previous episode. The antibodies bind to particular pathogens, giving a visible result that confirms their presence. It's important to note that not all plant problems are caused by pathogens. Plants also require specific mineral ions from the soil, and deficiencies can sometimes produce symptoms that resemble disease. For example, plants need nitrate ions to make proteins necessary for growth. When nitrates are in short supply, plants often exhibit stunted growth, a symptom that could be mistaken for disease. Similarly, magnesium ions are essential for making chlorophyll, the green pigment needed for photosynthesis. Plants lacking sufficient magnesium may develop yellow leaves, chlorosis, and have reduced ability to photosynthesize. This yellowing might be confused with symptoms caused by certain pathogens. Now, let's explore how plants defend themselves against pathogens and pests. Unlike animals, plants can't run away from threats, so they've evolved sophisticated defense mechanisms. Plants have several physical defenses that act as barriers against pathogens. Most plant leaves and stems have a waxy cuticle that provides a waterproof barrier, making it difficult for pathogens to enter. Beneath this waxy layer, plant cells are surrounded by cell walls made from cellulose. These tough structures form another physical barrier against pathogens that manage to get past the waxy cuticle. Many plants, especially trees, have layers of dead cells around their stems. These form bark, which acts as an additional barrier to stop pathogens entering. If damaged, these outer layers can fall off, removing potential infection sites. Plants also employ chemical defenses. Some plants produce antibacterial chemicals that kill bacteria attempting to infect them. Examples include mint plants and witch hazel, which contain natural antimicrobial compounds. Other plants produce poisons that deter herbivores, organisms that eat plants. Tobacco plants, foxglove, and deadly nightshade all produce toxins that make them unpalatable or even dangerous to potential predators. In addition to physical and chemical defenses, plants have evolved mechanical adaptations to protect themselves. Some plants have adapted to grow thorns and hairs on their surfaces. These structures stop animals from touching and eating them by making the plant difficult or painful to consume. Other plants have evolved to drop their leaves or curl when touched. This response allows them to prevent being eaten by knocking insects off themselves or moving away from threats. Some plants go even further with mimicry as a defense strategy. For example, the bee orchid, Ophrys apifera, has evolved flowers that closely resemble female bees in appearance and even produce similar scents. This tricks male bees into attempting to mate with the flower, facilitating pollination in the process. Another fascinating example is the dead nettle plant, which closely resembles the stinging nettle but doesn't actually produce stinging hairs. By mimicking a harmful plant, it deters herbivores without having to invest energy in producing actual defensive structures. Some desert plants, like living stones, Isoaceae family, have evolved to look almost identical to the rocks and pebbles in their environment, making them nearly invisible to herbivores searching for vegetation to eat. While plants may seem passive, they have sophisticated systems for detecting threats and defending themselves against a hostile world.